Introduction to Vision and Visual Processing. What does it mean to see? At first, the answer seems obvious. Light enters the eye. The retina detects it. The brain processes the information. And we see the world around us. But that simple explanation hides an extraordinary amount of complexity. Because seeing is not simply a matter of having good eyesight. We can have excellent visual acuity and still have difficulty processing visual information. We can recognize an object, but struggle to find it in a crowded environment. We can read the words on a page, but find reading exhausting. We can see a face, but have difficulty interpreting what that face is telling us. We can see movement, color, contrast, and depth, but the information reaching consciousness may not always be organized in the way we expect. This course is about that difference. It is about vision and visual processing. The purpose of the course is to examine vision as a complete biological information processing system. We will begin with something that most of us take completely for granted: light. Before we ever see an object, light has to reach the eye. That light is modified by the environment. It passes through the optical structures of the eye. It is focused onto the retina. The retina converts photons into neural signals. Those signals are then processed through increasingly complex neural pathways. And eventually, somehow, information becomes perception. But there is an important point here. The brain does not receive a photograph of the outside world. It receives signals. Those signals are incomplete. They are transformed. They are filtered. They are compared with other signals. They are influenced by timing, contrast, movement, color, attention, previous experience, and the state of the nervous system. The visual world that we consciously experience is therefore the result of a remarkable biological processing system. And that system is what we are going to explore. For generations, the clinical assessment of vision has placed enormous emphasis on a relatively simple question. Can you see the smallest letters? Visual acuity is extremely important. But it measures only one part of visual function. Imagine two people who can both read the smallest line on a conventional visual acuity chart. Now place both people in a busy shopping center. One person moves through the environment comfortably. The other finds the visual information overwhelming. Faces, signs, movement, reflections, patterns, and changes in illumination compete for attention. Or imagine two people reading the same page. Both can identify every letter. But one reads fluently while the other loses their place, skips lines, experiences visual discomfort, or becomes exhausted after several minutes. Their visual acuity may be identical. Their visual processing may not be. This distinction is fundamental to everything that follows. To understand visual processing, we need to follow information through the visual system. We will examine the optical system of the eye, including the cornea, aqueous humor, crystalline lens, and vitreous. We will examine the retina and its different cellular layers. We will look at the photoreceptors, the rods and cones, and ask what information they actually provide. We will examine retinal ganglion cells and the pathways carrying information towards the brain. We will explore the lateral geniculate nucleus and the major pathways associated with magnocellular, parvocellular, and corneocellular processing. And then we will follow information into the visual cortex. But we will not stop at anatomy. Because knowing where a signal travels does not necessarily tell us what happens to the signal along the way. We need to consider what happens to information as it is transformed. How is contrast extracted? How is color represented? How is movement detected? How is spatial information organized? How is timing represented? How are different signals combined? And perhaps most importantly, one of the most important lessons in visual science is that perception is not simply a direct recording of reality. The same physical stimulus can produce different perceptual experiences. A color can appear different depending upon its surroundings. An object can appear to change size depending upon context. A stationary pattern can appear to move. A perfectly normal visual image can become ambiguous. And information that is physically present may not necessarily become consciously available in the same way. These phenomena are not simply curiosities. They demonstrate something fundamental. It takes incoming information and constructs a representation of the environment. That construction involves multiple stages and multiple forms of processing. And when processing changes, perception can change. This provides an important framework for understanding many visual experiences that are difficult to explain using conventional measures of eyesight alone. Visual processing affects almost everything we do. Reading requires the precise coordination of spatial information, eye movements, attention, timing, and visual recognition. Driving requires the interpretation of movement, depth, contrast, distance, and rapidly changing visual information. Sport requires prediction, tracking, and extremely rapid integration of visual information with movement. Learning requires the brain to extract meaningful information from a constantly changing visual environment. An ordinary social interaction involves visual information too. Faces, gestures, eye movements, body position, and changes in expression all carry information. This means that visual processing is not an isolated specialist subject. It is part of everyday human function. And when visual processing becomes inefficient, unstable, distorted, or poorly integrated, the consequences may appear in places that do not initially look visual at all. A person may simply appear distracted, or clumsy, or slow, or uncomfortable, or unable to concentrate, or unusually sensitive to particular environments. The challenge is determining why. Throughout this course, we will repeatedly return to one principle. A symptom does not necessarily identify its cause. Difficulty reading does not automatically mean that the problem is located in one particular part of the visual system. Visual discomfort does not have a single explanation. Poor tracking does not necessarily have one cause. Sensitivity to light does not automatically tell us why the person is sensitive to light. The same outward behavior can potentially arise from different underlying mechanisms. That is why assessment matters. And it is why we need to understand the system before attempting to interpret its failures. Rather than beginning with a label, we will begin with the biology. Rather than asking only, what does the person do? We will also ask, what information is the nervous system receiving? How is that information being processed? And where might processing differ from what we normally expect? During this course, we will progressively build that understanding. We will explore the optics of the eye, retinal physiology, phototransduction, color vision, contrast, spatial and temporal processing, eye movements, visual pathways, cortical processing, magnocellular and parvocellular systems, dorsal and ventral processing, visual attention, pattern recognition, depth and motion, visual illusions, visual stress and discomfort, and the relationship between visual processing and other sensory systems. We will then move from understanding to assessment. How can visual processing be examined? What can conventional optometry tell us? What might require additional investigation? How can we distinguish between different possible explanations for the same visual complaint? And finally, we will examine management, lighting, lenses, filters, contrast, the visual environment, and ways of modifying the visual stimulus reaching the individual. This course is deliberately designed for more than one audience. There are two options, but we will use the same videos, but explain things in either a way suitable for non-experts, a way for professional use. The expert will also have a book with the script and references. For professionals, it provides a deeper examination of the mechanisms underlying visual function and visual processing. For parents, it provides a way of understanding what may otherwise seem like confusing or contradictory behavior. You do not need to begin with an advanced knowledge of neuroscience. We will build the subject progressively. Some sections will be technically demanding. Others will be deliberately simple. The objective is not to remove the complexity. It is to make the complexity understandable. So, when we ask, "Can this person see?" we are asking an important question. But it may not be the most important question. We should also ask, what are they seeing? How stable is that information? How accurately is it being processed? How efficiently is it being integrated? And ultimately, how does the visual system transform light into experience? That is where our journey begins. Welcome to Vision and Visual Processing.